Okay, so yeah, my name's Gary Groves. I'm a PhD student hosted by SAMS, which is part of the University of the Highlands and Islands. Work closely with UHI Shetland as well. That's where most of my work actually happens. So I work with these devices. They're called Imaging FlowCytobots. Basically, they're flow cytometers, but they capture images of all the particles which pass through them. So they're primarily designed to catch images of phytoplankton, and particle size, we're looking at anywhere between 10 to 150 microns. It does catch particles lower than 10 microns, but they tend to be quite indistinct and difficult to work with. They can sample, I think, up to about 30,000 images per hour, depending on how much we actually see in the water. We tend to find that our samples are between sort of 2,000 and 8,000 during sort of the height of the phytoplankton season. They can sit in the water for up to 6 months at a time, working away. They have kind of cleaning cleaning processes, which keep them from biofouling internally. And they work 24/7, so they kind of work round the clock. They take a 5 mil sample every 20 minutes and then process that. So we currently have 2 of these devices in operation. We have one deployed at an aquaculture site that's managed by Scottish Sea Farms. That's at a location called Cold Deep, which is in Shetland. And that's been in the water since February 2023. And so far, that's captured about 76 million images. So that's a lot of image data it's working with. And we have a second device. This one isn't actually in the water. It has seawater pumped to it. And that's deployed at the UHI Shetland campus in Scalawa in Shetland. And since about 2023, that's captured 43 million images. So it's not as active a site as the Cold Deep site, but still getting plenty of image data. So these things sit in the water, they take the samples, and then they upload the sample data to some cloud storage. And sorry, my animations are all slightly out of order. And then we have an AI classifier which looks at all of those images that have been captured by the device, and it tries to sort them into one of 77 different classes. So we try and capture all the kind of dominant phytoplankton classes which are present in the water in Shetland. And it uses a multi-axis vision transformer architecture to do that classification. So I'm not going to talk about that today, but I think I've got a talk coming up at the MASTS ASM. So if you want to hear more about the actual classifier and the wider project, then yeah, come along to that talk. And then once it's looked at the images, what we want is counts. So we want to enumerate the different genus or species in the water and publish those online. So really the project's aimed at monitoring harmful species and providing sort of timely alerts to aquaculture managers. So we have, this is an example of what the IFCB data looks like. So it's grayscale images. You can see there's a real difference in size. Hopefully you can see my cursor. Quite large clusters or chains down to kind of very tiny individual particles. This is a relatively small sample of just over 2,000 images. Okay, so we have a slight issue when it comes to the classifier. So the classifier is great at telling you what's in an image. Yeah, it's kind of does a good job, but we have a slight problem when it comes to these chain-forming diatoms. So we've got four example species. These are probably some of the most prevalent harmful species that we see in Shetland. We've got Chaetoceros, which you can see in the top left. Irritates fish gills. You can see all these kind of spiny setae protruding from the different cells. That kind of irritates the fish gills when they're in high volumes, and it is a very predominant species. And Flaciocera as well affects kind of the gill health in the same way. Then you have the top right here, we have Pseudo-nitzschia. So this is a domoic acid producing genus. And if it's ingested by shellfish, you can have toxin buildup in the shellfish, and that makes it unfit for human consumption. It can also have an impact on fish health as well, because there are toxins which affect fish health. And skeletonema as well. We just see this in such high quantities in the water that it becomes a problem. So what we need to provide to sort of the aquaculture managers is how many of these cells are actually in the water. So when the classifier sees one image, it will count this as one. So there's one Chaetoceros, but actually we can see there's multiple cells in the image, Thalassiocera even more so. So what we wanted was a way of counting the number of chains in an image as well. So I developed a chain counter. So this is kind of occurs after the classification step. And if the classifier sees one of these 4 genus, which are typically chain formers, it will then pass that image to the chain counter, which will then try and count the number of cells in the image. Okay, so developed this, we call it the DCCM, which is the diatom chain counter model. So it's based on the YOLO architecture, you only look once. We chose this really because it's very quick. It was easy to use in terms of sort of the coding, the libraries available and the support available, and it's really good at actual image detection as well. So I developed 4 separate models, one for each of the target genus, rather than a single model to try and capture everything. So we knew the classifier would be doing the classification step. So having a separate model for each target kind of made sense. There are different size variants of the architecture as well. So we tested each one of those. So just in relation to how many sort of processes it performs when sort of training and doing the inference. And we trained this on 1,000 images with an 80-20% train and validation split. So it uses 20% of those 1,000 images to actually check how it's performing at each epoch. And there are further 500 images which were set aside as a test set. And we manually counted, or I manually counted, how many cells I could see in each of those images, and then we would use that to compare it to how many the DCCM saw in each of those images. Okay, so just a quick comment on intersection over union. So the intersection over union metric, you can see is basically how much the bounding boxes or the detected bounding boxes overlap. So the intersection is where they meet, and the union is the total area they cover. So where you have kind of a high overlap, you have a high IOU value. Where you have a very low overlap, you have a low IOU value. So this is actually used during the training of the image detection algorithm. And it's used as kind of a metric to see how close it was to kind of the ground truth, which is the annotated data which we provide it. But I was more interested in how we could use it at the object detection stage. So you'll probably see from some of these images, we have chains where the cells are nicely spaced apart, but then we have chains like Chaetoceros being a prime example where all of the cells are very closely packed together. So this kind of like gave us different problems depending on which particular species or genus we were looking at. So we ran the inference multiple times against the training data and compared that against the test data, and then compared that with the counts, the human counts. And we tried to find what the best IOU value was for each of the different genus we were looking at. So for skeletonema, actually a really low IOU value kind of worked pretty well. You can see even as low as 0.025, so very little overlap was allowed. Because when we saw these cells, they were quite naturally spaced apart. They tend to have this kind of, you can almost see it in the image there, this kind of structure between the cells, which kind of binds them together. Chaetoceros, on the other hand, was problematic because the cells are very densely packed. You can see in this example, some of the cell itself or some of the chain itself is actually out of focus, makes it almost impossible to count for a human or a machine. But we found sort of much higher IOU values kind of worked well with Chaetoceros. So comparing the DCCM counts to the human counts gave us that kind of benchmark. So once we kind of picked the best performing models and the best performing IOU value, we kind of ran this against a year's worth of historic data. So we took the 2024 data from Scallower and Coal Deep, and we looked at, what's the difference between just counting an image as a single instance or counting all of the individual cells in that instance. So Chaetoceros was probably the most markedly different. We saw a kind of 7-fold increase in the number of individual cells we were detecting, which is kind of makes sense because Chaetoceros forms quite long chains with densely packed cells in them. Thalassiocera, likewise, we saw a big increase. Not so much for Skeletonema, and Pseudo-nitzschia was a bit of a puzzle as well because we didn't really see long chains of Pseudo-nitzschia, so we didn't really see a big increase in the number of cells versus just the images. So just some graphs showing the comparison. So Chaetoceros at Coal Deep, you can see the trace in red is just the image data or the entire chain, whereas the cells, and you can see there's a big increase in the number of cells. So for the Scallower data, we had an additional data source here. So Scallower is a site which is part of the Scottish Coastal Observatories program. So there's been phytoplankton monitoring happening there since, I believe, 2002. And they take weekly samples. The sample point is about 300 meters away from the IFCB intake point. So there is a slight difference in terms of location. But we can see in the samples that we did see a correlation somewhat, except maybe for this bit here where there were more actually seen in the microscope samples than the IFCB detected. Pseudo-nitzschia as well. I'll come to the second slide. So that's more interesting. So here's where we saw the really big difference. In the microscope samples, way more was observed in those than we saw in the IFCB counts, even with that chain counting model applied to those. And I'll come back to that at the end when we think why that might have been happening. Skeletonema, there was an absolutely massive bloom of Skeletonema at the Coal Deep site back in April last year, and that was confirmed by the Scottish Sea Farms team, who basically said they had a massive bloom at that time. We saw that in the IFCB. And you can see there's a slight mismatch in terms of kind of the microscope counts and the IFCB counts. We did see a few peaks in the microscope, which we didn't necessarily see in the IFCB. And Thalassiocera as well is another example where we saw a massive increase in the number of cells. And again, there was some match between what we saw in the IFCB and what is seen in the microscope. So just to kind of sum up, when we were comparing it with the human counted test data, we saw kind of a 95% plus accuracy for the Thalassiocera and Chaetoceros when we counted using the chain counter and human counts. And that did increase to 99% accuracy for Skeletonema and Pseudo-nitzschia. I think the advantage of those cells, they were always quite evenly spaced, whereas Chaetoceros and Thalassiocera, you often see quite spiraling cells, sometimes in and out of focus, sometimes self-obscuring. The unexplained difference between manual counts and automated counts, particularly for Pseudo-nitzschia at Scallower. Interestingly, I was talking to some other IFCB users, and they had experienced the same thing, although they hadn't run the chain counter. But kind of the hypothesis is that the larger chains are actually getting filtered out. So we have a 150 micron pre-filter before the samples are taken into the IFCB. So there's a high chance that any chains over that size just aren't going to be passing into the IFCB, so we don't see them. Future applications for the chain counter, looking at possibility of counting other phytoplankton chains. We picked these original 4 because they were the most impactful. They were kind of the most suited to this approach. There's possibility to kind of have that for other genus as well, and also for other instruments as well. There's the FlowCam, which is very similar to the IFCB, but is a desktop-based machine rather than an in-situ machine. There's no reason a very similar model couldn't be developed for FlowCam data. And possible refinements for the model as well. So we were just counting instances of a cell. but it might be that we can actually look for dividing cells versus non-dividing cells, or this is an example of an oxospore cell, which is part of kind of the reproductive cycle of Thalassiocera. So we could train our model to actually look for cells which are at these maybe slightly different stages of the life cycle. So we have got a paper that's been accepted by the Journal for Plankton Research. The amendments have been done. It's just been sent back in, so we're just waiting to hear the final decision. So that'll be coming out soon. Just a thank you to the team. As I say, it's a very small part of a larger project. So if you want to hear more about the larger project, I think I've got a talk at the MASTS ASM coming up very soon. And thank you very much for listening.